Hello friends, welcome to BMH Learning. This video is about DNase or DNase1 hypersensitive sites sequencing, also called as DNase1 food printing assay. It's an in vitro method used to identify the specific site of DNA binding proteins. It not only finds the target protein that binds to specific DNA, but also identify which sequence the target protein is bound to. This technique can be used to study interactions between proteins and DNA both outside and within cells. Its principle is based on the fact that a protein bound to DNA will often protect that DNA from enzymatic cleavage, and the enzyme used is DNase1. The cleavage pattern of the DNA in the absence of a DNA binding protein, typically referred to as free DNA, is compared to cleavage pattern of DNA in the presence of a DNA binding protein. If the protein binds DNA, the binding site is protected from enzymatic cleavage. This protection will result in a clear area on the gel, which is referred to as the footprint. Strategy: DNA fragment containing protein binding sequence is extracted, amplified, and labeled at one end of the double helix using polymerase chain reaction technique. Labeled DNA fragments and DNA binding protein along with DNase1 are mixed in a test tube. DNase1 will cut at random locations in a sequence-independent manner. In another test tube, labeled DNA fragments are mixed with DNase1 without DNA binding protein. This is used as standard to compare the results. DNase1 cuts the DNA fragment present in both test tubes, but no cuts are made at the specific region of DNA where proteins are bound. Protein has protected the DNA binding site from DNase1. DNA fragments are separated by polyacrylamide gel electrophoresis and visualized using autoradiogram. When compared with the standard, missing band, that is footprint, indicates the protein binding specific DNA sequence. Comparison of both samples reveal footprints or protein binding sites. This was all. Thanks for watching.